So there's two basic kinds of things that people do from beta images, and they then have all kinds of consequences for everything that happens next. And they are univariate analyses. I sort of want to do a little bit about each to set them in relationship to one another so that we understand a little bit about what the two kinds of analyses are, and then we can go deep into depth about either or both one at a time. Okay, so a univariate analysis typically asking some combination of two questions about neural activity. How much neural activity there is in response to your conditions, and where that neural activity is in response to your conditions. Those two questions are related, or at least they're not perfectly separable, but that's the two kinds of questions that you're asking with a univariate analysis. Okay. So for the how much question, we're going to assume temporarily that we've solved the where question. And then we'll go back to the where question. So the standard thing that you want to know in a how much question is, let's say there's some brain region I already know I care about, like the FFA. That's assumed I've solved the problem of where is the FFA in the brain. Another version to think about this is, for example, what if I care about the amygdala? If I'm interested in the amygdala, I solve the where question with an anatomical image. So I look at the anatomy of the brain. I can see the amygdala, which is a thing, right? It's a particular place in the medial temporal lobe, so I can see that thing, the amygdala, and that solves the where question. I'm looking there. Okay, is that clear? So then I know where I'm looking. And the question I want to know is, how much does that brain region respond in my experiment to my conditions? Right? Okay, so usually those brain regions, if that's the amygdala or if that's the FFA, they're bigger than a voxel. And so the how much does this region respond question, what that means is the set of voxels that are inside this region, how much do they respond on average? That's the simplest version of that question. And so the simplest answer is I take all the voxels that are inside that region, and each of them has a beta value, right? And I average them across all the voxels inside the amygdala. At the moment I'm still doing this. This is per person. Okay? So I found the amygdala based on anatomy, or the FFA based on prior knowledge, or whatever. I found the brain region I care about. Then I take all those voxels that are in there, and I take their average beta value for condition A and the average beta value for condition B, and that gives me a difference. Right? Okay. That difference of the betas is also called the contrast value, or the con. But that's just the name of it. That's the name of the value of the difference of, right? So now I know a number. Right? The amygdala for conditions A and B, the contrast value is 0.5, which is just as unhelpful as beta values were, right? Because that's beta value minus another beta value in arbitrary units, right? So now we just have, it's literally minus. So some people think there's magic here. There's no magic. That's literally a subtraction of two numbers. You can do it by hand if you want. Okay? So now we get the answer is 0.5. Okay. That is particularly informative. Is 0.5 big? Is it small? That's Veronica's question. We have absolutely no idea. It depends on how you modeled your data, right? But one thing you could ask is that consistent across people? A greater than B in the amygdala in every person I measure, right? And so that's in one person, and so now I could do the same thing in a different person's brain. Their brain, in their amygdala, right? And so one thing I could do is ask about this contrast value. I could ask whether it's consistent across people. Is that clear as a question? And since that just two numbers per person, right? So you have two numbers per person, a big row of two numbers. The statistical test you would probably do is a t-test, yes, which you can also do by hand. I have done t-tests on brain data by hand this way, right? This is not fancy at all. And, right, so you say if the t-value is big enough that the chance of getting that, if there's no difference between A and B is less than 0.5, then I accept that there is a reliable difference across people in the amygdala's response to A versus B. So that's a question, right? When we did this, we said, we think maybe it's interesting that A is greater than B in the amygdala. And you're saying, how do you know if it's right? Maybe that's for some totally boring reason, or maybe that's true everywhere in the brain, not just the amygdala. Maybe it's much bigger other places. And this analysis can't tell you that at all, right? So far, all this analysis can tell you is whether the difference is consistent in the amygdala. It can't tell you anything else. And because of that, these days it would be very rare to do a paper that did just this. So again, I'm trying to work us up through what is each piece of the analysis, and then we can see the context in which they occur. But yeah, so definitely if you only looked at the amygdala and you only did one t-test, it would be unusual for you to be able to publish that as a paper. Okay, but so this is, again, this is the simplest thing you could do in a univariate analysis where your question is how much. Right? Average betas here, that's averaging across voxels within a person. Right? Statistics are because we do it across people. The t-test is across people, which means that the signal is the average difference between the two conditions across people, and the noise is the variability in that difference across people, right? So one thing to note about that analysis is that the variability in terms of how well the model fit the data is gone, right? In this analysis, we don't know how well the betas fit the original data. There's no noise per person in this analysis. All we know is, in the end, the beta was different, was higher for A than B consistently across people, right? For regions, once you fit betas with decently comparable data, across subjects, the distribution of the betas don't violate any of the assumptions you'd make for a t-test in general. Yeah, and whether or not averaging these betas is a good way to represent the activity in a region, that totally depends. And there's a lot of, yeah, that's a complicated question. So the simple question is yes, if you do this procedure, then the across subject measures, it's reasonable to do a t-test over them. And we'll come back to what's inside this average many times, because averaging the betas per region is only one of the things you can do with them, and we're going to do a ton of different things. Yeah. So this is the oldest thing to do. It was the first thing people did. It was the simplest thing to do, and it's worth understanding. But it is definitely not the only. In fact, that's what I said. What you do with beta values is fMRI analysis, right? This is one option, and it's the simplest thing, assuming you've solved the where problem, and your question is only how much, right? So there was a good 10 years there in fMRI data where that was a sufficient question, the question how much activity, because then all of the excitement in fMRI is about designing A and B well, so that it's really interesting to know whether in some place the response to A is greater than B, right? And finding where to look, right? So for 15 years of fMRI, most of the interesting stuff was in thinking about where should I look And what experiment should I design such that it's interesting to know whether that brain region responds more to one of my conditions than the other? Is that clear? And so that became questions like, are there selective brain regions for specific jobs, right? The whole research program to say there is an FFA that is selective for faces was all of this form. Have an idea about where you're looking, and then design conditions so that you can test the hypothesis of selectivity. The EVS beginning research program, are there brain regions that respond selectively for language, not other cognitive tasks, has this form, right? Not just A and B, sometimes it's A through Z, right? But the question of how much you respond to different conditions, the whole conversation about selectivity was in this form. Pick a brain region that you're interested in, solve the where problem somehow, which I'm not telling you how. Once you know where you're looking, average the activity to each condition and ask which conditions get bigger responses and which ones get smaller responses. And then there's a big fight about the meaning of that difference, right? Is that sufficient evidence for selectivity? What does selectivity mean, right? So those kinds of questions about the meaning of the data, that's totally interesting. I was not planning to talk about it. All I'm planning to tell you about is the concept in the analysis, right? And there the concept is, you know where you're looking, you can average the betas to those responses, and then you can ask whether there's a reliable difference across people in the size and direction of that difference. Another whole interesting controversy, actually, is how big does that difference have to be important? That was, like, I don't know if you guys have heard that Nancy Camosher and Isabelle Gauthier had a famous, like, 25-year-long fight. It's about, is a big difference more important than a small difference? I'm not kidding. That's actually what the fight is about. 25 years later, they're still fighting. Both differences are significant. One is much bigger than the other. Is a big difference more important than a small difference in understanding a brain region's function? That's the kind of thing people fight about. Both. It is in both. It's a big difference in betas, but it's actually also a big difference in percent signal change. Yeah. It's a robust big difference, a for real big difference. And actually part of why it's a fight is because Nancy cared about the for real size of things and other people cared about the statistical significance, right? So bear in mind, a t-test, right, you can get a highly significant result in the sense of it's very reliable across people that's also very small, right? If A is just slightly bigger than B in every single person, the t-test will tell you that's significant, right? And so then there's a question of, okay, it's significant, it's reliable. Is it also big and important? And that's the kind of thing you can fight about for a lifetime. Yes, that's an empirical fact. You can fight about that for a lifetime. Okay, so let's do a little bit on where, and then we'll break for lunch. Okay, so I said this is if you've solved the where problem. You've solved it anatomically, or you've solved it based on prior knowledge, or somehow you know which group of voxels you want to count as a region. That, by the way, involves a philosophy of regions, which we can also talk about. Are there regions? That's a religious commitment that some people have and some people hate. There are religions of hating regions. So here we assume there are regions and we study their average responses. Now, what if you don't know where the difference might be? So for example, you think there might be a brain region that responds more to faces than scenes, but you don't yet know where it is. How should you find it? Okay? So now we're going to go back to our 3D data where we have a beta value per voxel. Right? So now, in every single voxel, the same analysis. Right? Average beta, this is just actual beta. Right? That's the beta value in that voxel, right? This is now on value in that voxel, right? And so we can take these two images, and if we subtract them, literally A minus B, we have a new three-dimensional image, which is the contrast image, which is the value at each point of the contrast value in that voxel from the difference of the two. Is that clear? So literally at every point, subtract the value of B from the value of A, you get the subtraction, which is a con value. Comprehension question. Can con values be negative? Yes. Is that clear to everybody? If the beta of B is bigger than the beta of A, when you do A minus B, you get a negative value. Yes? Great. OK, so that tells you the size of the difference at every position. Right? And there's some things you can do with just that. But actually, often what we want to know is, how does that size of the difference compare to the noise in that voxel, or the noise in that model, the unexplained variance, right? Because what we're interested in is, are there any places where the response to condition A is big, the response to condition B is small, and that difference is big relative to the noise? If it's super, super noisy, then maybe the reason that one beta is big and one beta is small is just the general principle underlying all statistics is that what you're looking for is a signal-to-noise ratio. The signal, the difference between conditions, how big is that relative to the noise in the thing I measured it in? Right? And so now here, what we want to know is, we don't have any other participants, right? This is one person. We have one voxel, the beta for A, the beta for B. And we want to know, is there anywhere where that difference is big relative to the noise in that voxel? Does that make sense? OK. And so then, because it's here in the unfit variance, we don't have it in the beta image. But we could go back to the data and ask, how well did our model actually fit the data? Right? So at every voxel, we know what our beta values were. But we could also have kept track of and how much unexplained variance was there after we fit that model. Right? Does that make sense? Because as Todd says, different packages do this differently. But in addition to knowing the con, right, for each voxel, so that is the difference of the betas, we can also know how much unexplained variance was there in that voxel after we fit those betas. Right? And so what we might actually want to know is, where is the con value big and the unexplained variance value small? Right? That's going to be a place where you know the difference between A and B. You know a lot about what's going on for this voxel in this task. It's basically a t-test. Well, in some cases, it's literally a t-test. T-test, then the image you produce is called again, per person. Big cons, small unexplained variance. That's a big effect relative to the unexplained noise in that voxel. That's a place where the difference in the signal is large compared to the noise that you can't explain. Yep. And so a T-map has a value at every voxel. Can T values be negative? T values can be negative. They can be large positive values. They can be large negative values. The null distribution is near 0. Effectively signal to noise. OK, so now we have another three-dimensional image, which is at every voxel, the T value, the size of the contrast A minus B relative to the unexplained variance in that voxel over time. Right? So for this participant, yeah? Are people with me? OK, so we're going to do one more step, and then we're going to break. OK? For anybody who knows this, at second-level statistics, we use this image, not this image, and Todd is asking why. And let's come back to that, because we're not doing second-level statistics. We're doing first level. This is a good question. So let's just make clear that everybody knows what our images are. We have a beta. We have a beta per condition. We have a con for any difference between conditions that we're interested in, right? So con relates different betas. And we translate to know whether those differences are large because we did a good job fitting a real difference in our data, or are large because our data were so noisy that you could fit absolutely anything to them. Then we correct for the unexplained variance. And so now we know the difference between the conditions that we were interested in, how big is it relative to the unexplained variance in that voxel for every voxel, right? Yes? And so one thing you could then ask is, okay, tell me where in the brain the t-values are large. That question asks, tell me which voxels had a big contrast value relative to their unexplained variance, right? In order to say what's big, you have to set a threshold, right? So in order to look at a t-map, usually you don't look at every single value. You say, I just want to see the big values, right? Is this clear to people why? Because there's literally a t-value at every point. The question is not, is the t-value 0.0 or 0.1 or 0.2? Our question is, where are the big t-values, like bigger than 3, bigger than 6? Big, significant effect. OK, and so what we then do, almost always, is threshold this image. And what that threshold should be, there's a whole bunch of questions there. But a standard generic practice would be to look at your data where the t-values are pretty big. Therefore, the p-values are pretty small, like 10 to the negative 3. This is related to, but not the same as, the multiple comparisons problem that we will come back to. OK, but so then what that gives us now is a new image, which is a thresholded t-map, where we color voxels by their t-value only if they have an effect bigger than our threshold. Right? And so now everywhere where the effect is smaller than our threshold, we don't see anything. And everywhere where the effect is bigger than our threshold, we put color. That shows us, could be single voxels, right? Could be one voxel at a time where the effect is really big. Often, for lots of reasons, it's a set of contiguous voxels. And so there's a whole bunch of voxels all side by side where the t-value is large relative to this threshold. And that's OK. And then, for some purposes, this is the answer to the question. So in some papers, what you might want to say is, if your question was, where in the brain is there a bigger neural response to condition A than condition B, what you might publish is, for each person, a picture of their brain with colored blobs on it. And if that's what you publish, again, Nancy's first papers in the late 90s, early 2000s, one of the main figures is a picture of each person's brain with colored blobs on it. And this is how you get them. In the baby paper, we did the same thing. One baby, right, their brain, and colored blobs on it. If that's what you're publishing, you got them this way. That is relatively unusual now in adult standard fMRI analysis, that you publish each individual person's brain. But in weird populations like babies or people with brain damage or whatever it is, then that's what you might publish, each individual person's brain and a statistical map on their brain of where the differences were. Okay, and then also, obviously, you could use this answer as the input to this, right? So you could use one data set in a person to figure out where in their brain they responded to something, and then in another experiment measure how much they responded to something. And so you could use this answer as the input to this question. So that is what it is. So if somebody says they're doing an individual subject functional region of interest, what it usually means is they're using one experiment to find out where the voxels are that show some effect they're interested in, and using that as the input to another experiment where they ask how much do those voxels respond to some other set of conditions. And then this is the localizer, and that is the main experiment. Veronica's saying, look, if you scanned the whole brain and you found out where you were interested in, would you then keep scanning the whole brain? What a waste. Go back and scan just the part of the brain you're interested in, and now you can have higher resolution and so on. So no, we almost never do that for a bunch of reasons. One of them is this analysis almost always happens offline a long time later. So under some very specific circumstances, you might do a quick and dirty version of this while you still have the person in the scanner. but that's unusual and you have to be in some particular situation to be doing that. Much more commonly, you will not do this until much later. But so for example, back in the day, right, when it was normal not to cover the whole brain, if you knew you were studying visual cortex, you know it's in the back half, right? Like you might not be sure exactly where it is, but it's definitely behind the ears. And so that was why you could get away with scanning half the brain. You weren't going to not cover visual cortex if you scanned the back half of the brain. So under those circumstances, there were some situations where you might give up coverage for resolution or something. But I don't know whether people interested in specific subcortical structures, they might do that, right? So if you might use it, because an anatomical scan you do in the scanner, you can look at that right away. You can bring up the anatomical image. And so, for example, if you're interested in the thalamus, you might set slices that only cover the thalamus and never mind the rest of the cortex, and then have much higher resolution in the thalamus, which you can tell from anatomy. You don't need functional data analysis. Pardon? Yeah. Any part of subcortical, the subcortex is so much smaller than the cortex that if you're really interested in subcortical structures, then you might use the anatomical image to tell you where they are and make your slices just cover them and not worry about the rest of the brain. As I said, we do that with cerebellum, right? So we look for where the cortex is, we cover this cortex, never mind the cerebellum. So that's based on anatomy. But it's pretty unusual, and I can't think of a normal situation, only an extreme situation. I can't think of a normal situation where you do functional analysis and then figure. And as a very brief aside, one reason for that is ghosting. So it's if your slices, if the of the slices is inside the brain, not outside the brain, you get wraparound and all kinds of craziness. Anyway, that's a separate topic for acquisition. Yeah. This picture that I showed you, as I said many times, this answers the where question for a contrast in a single person, right? So if I ask the question A and B, let's say that's verbs and nouns, where in the brain is there a bigger response to verbs than nouns? In this person. Okay, then I have them do an experiment where they read verbs and then they read nouns and then they read verbs and then they read nouns, and I model their HRF. And I get an amplitude, which is my measure of neural activity for each condition. And I get the difference of those everywhere in their brain. And I get the size of that difference relative to the unexplained variance everywhere in the brain. And that gives me a T-map, which I threshold. So now I can say, here are the regions in that person's brain where the amplitude of the bold signal, and therefore the inferred amount of neural activity, was larger for verbs than nouns for that person. Okay, the most basic next question is that just that person? Or is that stable across people, right? And so the most standard thing you might want to do, just like we did up here where we said we want to do a t-test across people, where we ask whether it's not just that this one person's amygdala prefers faces to scenes, but that everybody's do. Here now we're verbs and nouns all over the brain. Is it just this person who has regions in these places that respond more to verbs than noun? Which could be something about that individual human or could be noise in that scan session. Or is this something that's true of people? Have we discovered the verb region of the brain in every speaker of English, let's say, right? And so to test that hypothesis, you have to know, is this representative of people? That is a question about the reliability of this effect across people. Yes? People are with me? OK. So there's a number of ways to do this, but I'm going to teach you the standard one, which is how do we ask whether this is representative of a group, right? And the standard way to do that is a whole brain group analysis, in which we ask whether this effect is not just true in one person, but reliable across people. Okay. There's two steps to this. One is, how do you do the statistics across people? And the other question is, how do you get the brains aligned so that it makes sense to do statistics across people, right? So to do a whole brain group analysis, we're going to do two steps. Okay, so do you guys have the the mental picture of what we're doing, we're going to try to come back to this kind of picture, but where instead of having statistics within an individual, we're going to have statistics across people. So we ask of every voxel, do you have a reliable effect across people, not just within a person? And then we're going to threshold that map and get another map that looks just like this, but is now representative of a group of people instead of a single individual person. But before we can do that, we need to know that we're asking that question of, so actually, let's do this, and then we'll come back. So we'll do step 2, and then you'll see why we needed step 1. OK? Is that clear? We're going to go the wrong way conceptually, because I think it will help motivate what we're doing. OK. So these are this Kahn image, but for a set of people, 3 different people. A minimum of 8 people. It could be more than 8 people, but it's never less than 8 people. Right? So for each person, we did this whole procedure. We ran the experiment. They read verbs and nouns. We fit a beta for the verbs. We fit a beta for the nouns. We subtracted the beta of the nouns from the beta of the verbs. And we got a Kahn image so that at each voxel position in each person's brain, we have the size of the difference between the betas for verbs minus nouns. Yes? Everybody's clear on that because that's what we did before? OK, so we did that for each person, and now we have that image for each individual person, right? OK, so that means that if we take a particular voxel, let's take this one. OK, so if we have the same voxel across each person, then now we have in this one voxel a Kahn value per person, right? Is that clear? And so we can average those values. And then we will get for this voxel, what is the average Kahn value? That's literally averaging. And again, you can do that by hand, right? Right. That's why we have problem 1. OK? So what I'm going to do is say, let's assume problem 1 is solved, and I'll show you what we do. Then we'll go back and think about how would you solve problem 1, right? Good. Is that clear to everybody? So this is exactly why I was like, hold on, I'll skip this and show you this, because as soon as they show you this, the question arises. But wait, you said that's the same place in every person. How do you know that? Right? OK, good. So that means you get the problem. Now let's assume I solved it. It's surely false. OK? Now, that is the same place in everybody's brain. Now we have a simple problem, right? In everybody's brain, we have a Kahn value, the difference of the betas, right? So here we could make a new Kahn image, which is the average, literally the average Kahn image, right? Then, from that, for every voxel, could look not just at the average Kahn value, but the variability in that Kahn value across people. Right? So remember, in our first participant, it was 0.5. In our next participant, let's say it's 1. In the next participant, it's 0.3. That's variability in the Kahn value, right? So we can take the average Kahn value relative to the between subjects variability. This is different variability than that one, right? Is that clear to everybody? When we did this map for an individual subject, we took the unexplained residuals from their time course. When we're doing this across subjects, we have dropped all that variance. It's not in our model at all. We took the Kahn images, which are the differences of the betas, and the variability is the variability between people. Yes? You take the Kahn value from each subject. You take the average Kahn value. You take the variance in the Kahn between subjects. And now, in each voxel, you can say, what is the size of the average size of the difference relative to the variability across subjects? And that gives you a T statistic again. Yes? Then we can do the same thing. We can apply a threshold. Ask, show me voxels in the brain where the T value is large. Now, that is that the average effect size across people in that voxels is big relative to the variability in that effect size, across people, right? The average effect across people is big relative to the variability across people. And there again, you could get full voxels, but maybe also sets of voxels in which the value is large relative to some threshold. Okay, I just want to introduce two words and a question, and then I'll answer your question. Okay, so one thing is just language. This is sometimes called a second level analysis. So sometimes you'll see people say, we submitted the data to a second level analysis or something. Whenever they say a second level analysis, what they mean is they did one analysis to get here per person, and then they combined the data per people in another analysis, and so they call it second level. So when people use the words first level and second level is within a person's brain, second level is combining across brains. That's one kind of language for it. This is not confusing if you call it a whole brain group analysis, because if it's a group analysis, it's a group analysis. The other thing people might say is that it's a random effects analysis, which refers to the fact that the variability is between subjects. When you do the t-test is a magnitude of the effect size relative to the between subjects variability. That makes it a random effects analysis, because the variability is the variability between people. Okay, and then the last thing I'm going to say is I didn't tell you how to threshold this. That's a whole question. We can talk a little bit about it now, but we could do a whole topic on how do you set the threshold appropriately. And whereas I said here, there's some standard practice for how you set the threshold, but it doesn't matter much. Here, the threshold does matter much. The reason is because very often, people are using this kind of image as the main result in a paper, right? That is very often what you will see published in a paper is a picture of this result. And so the question of whether or not, like how much statistical confidence we can have in that result becomes very important to understanding what claims are being made in fMRI and cognitive neuroscience. And so there have been big debates and big mistakes and drama and ad hominem attacks and naming names and controversies about this threshold because of the idea that if you set it wrong, you can publish false positives. Todd said, why do we use con images, not T images? Because the T images contain some information about how big the effect size was, and the con images do not. This is another question that we answered in that class N years ago. And the answer turns out to be basically that cons are better behaved statistically. They fit a better distribution. And for a random effects analysis, if the beta value was a bad fit, it won't be stable across people. And so the stability across people is the signal you end up looking for. I can go into more details on that if you want. I'm not fully prepped for that because I resolved it for myself many years ago and closed it off. But one thing to know about this kind of analysis is that it makes the power of your analysis much more sensitive to the number of people than to the quality of data per person. This is a thing that has been well studied, is that if you're going to do between subjects random effects analyses like this, that the power and robustness of your experiment is much more dependent on how many people you have than on how good your data is per person. So that if you had the same number of scan hours to distribute, you should cut the time per person in half and multiply the number of people by 2, and you will have much more robust statistics in a random effects group analysis for the same amount of data collected. Because, again, the effect size is relative to the variability, and so measuring that variability well becomes more important. Yeah. Okay, so, right. So when you are, not everything you do in fMRI is like this. But if what you think you're going to mostly be doing is a whole brain group analysis, then the answer is, yeah, more people rather than more data per person. So there's 2 questions that we haven't resolved. One question is, how do you align your brains? And another question is, how do you set a threshold here? And just to make clear why that's a problem, if we have 30 slices, and each slice, oh, I didn't say how big these are. Well, anyway, let's say we have 200,000 voxels, right? So if we do 200,000 t-tests, we certainly can't accept as a significant effect any effect whose probability of occurring by chance is 1 in 20, right? Because at 200,000 t-tests, you're bound to find a fairly large number of effects just by chance. So that's the multiple comparisons problem, right? The question is, if you collect 20,000 voxels, then how do you set your threshold for a significant result appropriately so that you see real effects but aren't overwhelmed by chance noise effects? That is the problem of multiple comparisons. And there are many possible solutions and a big debate about the most appropriate solution. And that's something we can come back to and have a whole topic on. Best way of setting your threshold for significant for the purposes of claiming there is a brain region that does X in a paper, right? Which is what often this image is for. OK, aligning brains. So Jin noticed this. I said, that blue voxel is the same place in those 8 people. She said, how do you know that? That is a very reasonable question. And so there's a big set of issues around how to solve that problem, how to make it such that when you go to do this analysis, you have the best shot that the blue voxel is the same place across the different people. The minimum thing that people do, sometimes called normalizing, sometimes called warping, it's called all kinds of things. The basic idea here is that we have an image of an anatomical brain. This is a template brain, and depending on which software package you use, you might have a different one, but it's usually it's a set of high-resolution anatomical images that have been averaged together. So it's like, you can think of this as a pretty good representation of the average brain. How many people here have heard of the Talairac brain? OK, so the Talairac brain was one person's brain that was scanned really carefully in Montreal about 20, 30, 40, 50 years ago. Does somebody know how many decades ago? Some number of decades ago, they scanned one woman's brain really carefully, and then they used that as an atlas. They said, if you want to say where you are in the brain, we'll name all the parts in this picture, look at your picture, and then figure out what it looks like in this picture, and that's where you are. So they sliced it in all the different directions, and they showed you this is what it looks like, sliced this way or sliced this way. And you would look, literally, you had a big book, an atlas, and you would try to find the picture that looked most like the picture you were looking at. On the picture would be names, and so you would say, OK, if I see this shape, OK, it's on page 92 that it looks most like that, and the label on that shape is the intraparietal sulcus, so I'm in the intraparietal sulcus. I actually remember doing analysis this way. That's how I did it. So you would and the books are beautiful, too. OK, so that is one person's brain, the Talairac brain. Great, 1967. So one thing you might wonder, one person scanned in 1967, is that the best representation we could possibly have of the human? I do know that her temporal lobes had degenerated to some degree. Yeah, and so there was some sense that maybe she was not the perfect specimen for all of humanity. So at some point later, people decided not to use her brain as the representative brain for all of humanity. And so various different templates have been constructed. The generic brain in SPM, I think, is the MNI 152 brain, which is 152 people scanned at the Montreal Neurological Institute and carefully aligned and averaged by hand. So in any case, so you should think of this as a picture of a brain, maybe some lady's brain, or maybe a decent representation of what brains look like on average. Okay, human adult brains, usually. The whole question about what to do if you're scanning kids or babies or people with only half a brain and so on. For now, assume we're scanning adults, and this is a picture of an average adult brain. Okay? So that's what the template is. It's a picture of what an average adult brain looks like. And then we have, for this subject, the anatomical image that we took for this particular subject, right? This particular brain. This particular person's brain. Okay? Now, because those two images have really high spatial resolution and pretty good SNR, what we can try to do is fit the best fit between these two images. So figure out for each point in this image, where's the point in this image that is most similar? There's a lot of different choices of what algorithm you use and what space you do this in. But pretty much all of them include at least changing so all the affine transformations, so that is expanding, contracting, stretching up, stretching down, right, rotating. So 3 dimensions of rotation, right? 3 dimensions of translation, and all the scaling, right? So basically you can think of, okay, to get this brain to look like that brain the best, okay, I need to smoosh it down, right, because it's too tall, and I probably need to lengthen it a bit, and maybe I need to turn it to the left, and maybe I need to stretch it a bit longer. Does that make sense what I'm talking about, what the affine transformations are? And so that is, you take the picture of the anatomical image, the picture of the template, and do all the turning and stretching that you need to do in the image as close as possible. Some then allow twisting, right, where you like bend the brain around a corner or something, and some don't, based on different preferences of software packages. So there's many different software packages that implement this step, where you try to take an individual person's anatomical image and figure out what is the mapping I need to do to map it into the space of the template image. And that is using the really high spatial resolution that can show you the difference between the cortex and the white matter, the structure inside the subcortex, the boundary between the brain and the peel matter, right? All those, in the image, all those dark and light boundaries, right? Every boundary where you cross a tissue boundary changes the brightness. That's what you're trying to map. And so the fact that this is very high spatial resolution and very good quality is critical to making this fit be able to be a really good fit. Some ways of doing this use the sulci in particular. So one way of doing this fit is first find in the individual brain all the sulci and make a map in the template brain of the sulci and align by the sulci, right? So that's one strategy for how you can do this. Others do pixel to pixel, so the whole brain simultaneously. There's many different algorithmic tools for doing this. And one thing you can do with your life in fMRI analysis is spend your time to figure out what is your preferred way to do this alignment. The more your conclusions depend on this kind of random effects analysis, the more important it is that you get your alignment correct for the reason that Jen said, right? If your random effects analysis is voxel by voxel, then having voxel be in the same place across subjects is more important. So we'll talk about other kinds of analyses you can do where that's less important, where getting this exactly right is less important. Okay, but 2 more things to say about this. The first thing, this is the person's anatomical image, but what we actually need to normalize is their functional data, right? And so in most normalizing algorithms, done in 2 steps. You align the anatomical to an anatomical template, and this is often registering. You register subjects' functional data to their own anatomical, and then you have the mapping, the 2-step mapping that tells you how to align the functional data to the anatomical template. Is that clear? The advantage is that this step, you assume the brain did not change shape or size between the 5 minutes in which you took the anatomical image and the next 10 minutes in which you took the functional image. That constraint means that this mapping is much, much, much more constrained, right? You don't scale it, you don't bend it, all you do is rotate it until you find the best fit. And that makes up for the fact that, remember, these data are much worse quality. They're lower spatial resolution, they're noisier, and so forth. And so it's very hard on these data to get the brains aligned really well. right, if you take two functional images, the data are so bad that your alignment will be very bad. But if you, aligning the functional to the anatomical with a rigid rotation, you can get that pretty right by knowing the constraint, that the brain is not allowed to change shape. And then you can get this one right by having a very high quality image. Two-step registration and normalization is the best way, using just what the image looks like, to get the brains aligned across subjects. So that's one way to do normalizing and aligning, is to use just the images, what the images look like, the pixel values, in order to create the alignment. And again, we usually do that in these two steps. Another thing that you can do is try to, once you've got a pretty good alignment across people, is try to improve it by knowing something about the function of the voxels, by knowing how they respond to different conditions. And that is what's sometimes called functional localization, which is to say, we'll trust this to get us close, but not perfect. And the reason is that we know for sure this process is not perfect. It definitely is not perfect. The reason is that, so how we know this, if you take, for example, a brain that's dead, map all of its functional regions, for example, based on cytoarchitecture, so it's anatomically defined areas, and then you do this process to it, and look, how does it align to another dead brain? The answer is, for example, that V1 only overlaps about 40% when you do it this way. And V1 is the easiest, best thing to find. So once you're in V2, it's like 20%, and for higher order regions, you can get almost no overlap at all using this procedure. So we know for sure. That's right. Well, it's for, yes, that's right. It's because the exact location and size of functional regions varies across people. And it varies in ways that are not visible in the images that we have, right? So if you look at, for example, relative to the sulcal markers, like even if you know where all the sulci are, the exact location of the same functional region, even if it's similar, just is different enough across that the best alignment you can do based on what you can see in an anatomical image, sulci, gyri, the gray-white boundary, like tissue structure boundaries, from what you can see in an anatomical image, even the very best alignment you can get cannot perfectly get functional regions aligned. Again, we know that's for sure for V1, and V1 is the easiest. As you go up to higher regions, you get less and less good alignment. And so that problem, which is, so you can improve it with better registration algorithms and better normalization algorithms, but you can't get rid of that problem. And so there's been interest always in not stopping with these two steps, but adding an additional step to use functional information to improve the alignment between subjects. And basically, there's two ways of doing that. They're very profoundly related, which I'm not going to talk about. One is functional regions of interest, which I talked about a little bit before. Other is hyperalignment. So both of those are tools for once you get your brains pretty close to aligned, based on the spatial structure you can see, adding functional information to match voxels to each other, not only that are in the same spatial location, but that also that seem to have a similar function in some way or other. Okay? So we can come back to what those are and exactly how they work, but just to give you the sense that that's the problem they're solving. Almost everybody, almost all the time, will have done at least normalization and rigid registration of the anatomical, of the functional data to their own anatomical. And some people will have tried to do something else to get the registration better. And then once you're done doing that, then you go on to these steps where you say, okay, now the data from this voxel, 52, negative 2740, right, that particular voxel, we're going to treat it a measure of the same brain region across all the different subjects and proceed with this analysis. Basically, what you're doing is treating it, instead of it at the voxel level, it's at the region level, which is slightly larger. But an ROI is a way of solving the registration problem. Yes, that's what it is. It is a way of solving the registration problem. ROIs solve two problems, registration and multiple comparisons. That's blowing my mind, kind of. Yay! I'm so glad to be blowing your mind. I'm sorry you don't understand me. But I will be happy to go through it again. But yes, what a functional ROI is doing is solving the registration problem. That's one of the things it does. It solves registration and multiple comparisons. And so once I find the voxels that are your FFA and the voxels that are her FFA, I count that as the same place in your brain, and I do random effect statistics across the data that came from it. Unit is not voxel anymore, but voxel was never an interesting unit, right? Voxel is a proxy for place in the brain. So all of this is a way of solving the problem, what is the same place in your brain and my brain? And once we have decided we found the same place, then we do random effect statistics on the in that place. And one way of thinking of place is voxel, which is frankly crazy. A voxel is a totally artificial thing. Another way of thinking of place is ROI. But all of these are different ways of solving the question, what is the same place in your brain and my brain? And what the way functional ROIs operationalize that, they say the three-dimensional volume that we collected subsampled from the true structure of regions, and therefore the alignment problem is an alignment problem of regions. And we solve the alignment problem of regions with functional data and some spatial anatomical constraints. And then we align the two subjects region. Okay. And now they're aligned, and we do random effect statistics on them. And you could have as many regions as you want. And so, for example, if you used resting state partialations to get 280 regions, then you'd have basically a random effects analysis in the space of 280 regions. And hyperalignment is exactly the same intuition, but continuous, not binary, for anyone who cares. Yeah, so in ROIs, you do this by declaring regions to be by a binary condition. They respond to A greater than B. In hyperalignment, you say the entire brain is mapped by a continuous parameter of its function, and so you fit each voxel to the most similar voxel in functional space. But it's exactly the same idea, which is you use the spatial structure of the brain for an approximate guide, and then some functional signature to get a more appropriate registration. Group analyses. Another topic is how to do multiple comparisons correction. So just so you know, those are two of the topics that were on my original list. Which means that that's basically univariate analyses, modulo the two big topics we didn't talk about, right? Univariate analyses ask the question where and how much, usually in these ways. Either within a subject where, or in a group where, and then once you know where you're looking, how much, usually by looking at the size of the contrast relative to the variance across subjects. So random effects analysis on the magnitude of activity.